First of all, thank you also for the opportunity to contribute to this very interesting webinar series today. So I will build a little bit on the introduction also that Miriam already has provided, and also give you some some overview of what we have been doing within IMI conception, especially the work packages 3 and 4. And I do want to emphasize that this is the result of the work of many people, many parties. I'm just speaking on their behalf here today. So what I will be discussing is the non-clinical and clinical approaches that we have developed and used to determine the secretion of medicines to breast milk. So in terms of IMI conception structure, I don't think I need to spend a lot of time here, but just mention you that we are here referring to work package 3, which were the non-clinical approaches for lactation, and then work package 4, where the actual clinical lactation studies and the biobanking was done. To start maybe with the clinical aspect, so in order to study the extent of passage of drugs into human breast milk, you can of course do clinical lactation studies. This is quite challenging because of course the population where you have to do these studies is a very special, a very vulnerable population. However, I think the good news is that within IMI conception, several of these clinical lactation studies have been done. Here you see the example of levocetirizine, also cetirizine. It's essentially the same drug where actually the transfer of these compounds, this drug has been studied during lactation in breastfeeding women. And so you see here a typical concentration time profile. So what you would typically also see in plasma, but in this case, you see it in milk, that you have a peak concentration that's being reached, and then gradually over 24 hours, the drug is disappearing again after this single dose of this drug from the milk. Of course, if this drug is appearing in the milk, that means that also the infant that is breastfeeding can get exposed. And that's, of course, the type of information that you want to generate here. What is very important in this context is that the protocols that you can use to generate this information, they have to be very well thought out, and you have to consider a lot of ethical aspects. This is just another example of a protocol that was recently published. It was in collaboration also with the group at the University Hospitals in Leuven. We called it Umbrella Study Protocol, which is kind of an umbrella protocol that was developed, which can be followed to study the transfer of different drugs in breast milk. This was a very, in a way, I could maybe say opportunistic approach. So we were able to get this approved by the ethical committee. That means whenever women were available and were willing to actually donate a few milk samples, that we could take the opportunity to measure the drugs in the milk and to have some information that in many cases for many drugs is otherwise not available. So please also go to this very recent publication if you want to read more about that. Before I dive into the non-clinical approaches, I just want to mention for the clinical lactation studies, what you can consider a primary outcome is the so-called milk plasma ratio. And it's actually the exposure that you have for the drug in the human milk divided by the exposure in the plasma, so the systemic exposure. So it gives you a relative idea how much exposure there is in human milk compared to the exposure in plasma. Exposure in plasma, which is commonly known in most, of course, for all drugs that are on the market. So, but if you have this milk to plasma ratio, you can make a lot of predictions about the exposure and can start to make assessment about potential safety issues. So what are then the non-clinical methods? And that's a bit the focus of work package 3. And the focus of this presentation will be a little bit more on that part, because that was also the part that I was personally more involved in. It doesn't mean it's more important, but I can just tell you a little bit more about that. And of course, we can build on that further during the discussion and the questions. So what kind of non-clinical methods can be used and how can they help to fill what you can call the data generation gap regarding medication safety during lactation? And then here in this case, we're talking, if we say non-clinical, we're talking about in vitro models. I'm referring back to what I what was just mentioned also in the previous presentation, where actually you can mimic also the blood-milk barrier with these mammary epithelial cells. You can use bottom-up in silico models. We call it physiologically based pharmacokinetic modeling, and that actually refers back to what Michael was also discussing, the biodigital twin concept. And then you can also use animals in an in vivo, complete in vivo setting. This is kind of the graphical abstract for these non-clinical methods. Of course, on the far right, this is the kind of information that we want to generate. As already indicated in the beginning of this presentation, you can use a clinical lactation study to generate that information. However, of course, that is quite challenging, and it will take still a lot of time to generate that information. And we should, of course, continue to do that. It will take a lot of time to generate that information for all drugs on the market. Essentially, if you dose mothers during breastfeeding with the drugs that they need, some of the drug may end up into breast milk. That's what you can measure in the milk samples. But you could also consider to extend your study design to also do opportunistic samples, for instance, in the infants after breastfeeding, and to see to what extent they have some of the maternal medication in their blood. So in order to combat to that information, indeed, you can take the clinical approach, but you can also take what we call two non-clinical pathways. The first pathway is what we call the in vitro in silico pathway. The in vitro model is what also was presented in the previous presentation, or at least in terms of concept, where you can measure concentration differences or the extent to which compounds are able to pass from one to the other compartment across a barrier of mammary epithelial cells. So the blood-milk barrier, you basically mimic that in vitro. And that kind of information can then be incorporated after scaling, after extrapolation in this physiologically based pharmacokinetic model, where you actually combine physiological information about the population. As for instance, you also describe, apart from the different organs, you also describe the breast milk compartment, and that compartment then directly feeds in, so to speak, into the infant PPK model. So in this way, you have a complete digital representation about the maternal and the infant physiology, and you can conduct, let's say, virtual clinical trials in these populations. And that would allow you also to predict this milk plasma ratio. Of course, you do need some measurements for drugs, at least in this point in time, via clinical lactation studies in order to be able to validate this approach. Another part that we also have explored as part of work package 3 in IMI conception are the in vivo animal models, where the Göttingen mini pig was selected as animal model. And I will just show you some data later on. Actually, this was actually quite successful in predicting the milk plasma ratios. The milk plasma ratio was determined in this animal and was quite predictive of the milk plasma ratio that had been determined for the same drugs in humans. Of course, we're still in the process. You see here also cross-validation to leverage as much as possible the advantages, but also the limitations are there, but the advantages of the different model systems to strengthen the predictive ability of these non-clinical pathways. So a little bit of text here to just give you maybe a little bit more background about what kind of molecules did we study. The focus was on small molecules, and the focus was really to quantitatively assess the passage of medicines across this blood-milk barrier. And in this way, then it was possible to assess the exposure to maternal medication. Important, of course, whenever you use non-clinical methods, that you can translate that to the human situation. What is the exposure in human milk? And then further on, what is the exposure in the infants? And that could be done with the in vivo animal model, but certainly also with the biodigital twin, with the PPK model approach. And the anticipated outcome of work package 3, and that's where we are more or less now, is really a proof of concept that we have generated for what we call a non-clinical platform, because you have different model systems for exposure-based medication safety during lactation. Initial verification, as is mentioned here, has already been done, because luckily, we do have some measurements from clinical lactation studies which can be really used to validate the approaches that we have developed. We have been in contact also with regulatory agencies trying to get qualification advice. It's a little bit of a long story where we are now, but I can definitely give you some feedback maybe based on questions later on. So in terms of compound selection, we have focused on small molecules, also molecules, drugs for model drugs for which clinical reference data were available, because otherwise you cannot validate what you're doing. We did look at diversity in physicochemical properties in terms of lipophilicity, to what extent these compounds were ionizing at physiological pH. In terms of pharmacokinetic or ADME properties, are these drugs mainly cleared by the liver or the kidneys? And this kind of diversity was built in the compound selection. And you can see here on the right, these are just the lists of the compounds. So most of the compounds that we have tested. For the in vitro and the PPK model, you have more compounds because they are higher throughput than the in vivo animal model, but still all the models have their advantages and limitations. So it was important to have them side by side. I'm not going to read maybe all the technical details about the animal model, but let's say the conclusion here for selecting this model system was that as compared to rodent models that were currently used or until now used for lactation, the Göttingen mini pig was really the most optimal choice in terms of in vivo animal model because of the physiology of the milk glands, which is much more comparable as compared to other species to the human physiology in that respect. The study design was to actually sample both the sows and the piglets for a period of about 3 weeks after also some a week of training. So this was quite an intense type of study, but also generated high resolution data for drug milk excretion. So also the kinetics really for these different compounds that were tested could be extracted like this. This is just an overview of the drugs that were tested in this in vivo animal model, including amoxicillin, venlafaxine, metformin, and levocitrazine. And you will recognize some of the compounds were also already tested in clinical lactation studies, which of course makes it interesting because you can compare afterwards. What you will see is that in most cases, and venlafaxine is an exception, the plasma concentrations are higher than the milk concentrations. In levocitrazine, it's maybe a little bit more break even, but for venlafaxine, the milk concentrations were higher than the plasma concentrations. So this was more of a lipophilic compound that is accumulating into milk. We also had the opportunity in these in vivo animal models to measure in the piglets, and what you see in actually for all the compounds, and that's actually good news. Of course, for drug use, medication safety during lactation, that even for venlafaxine, the concentrations that we found in the plasma of piglets, and that was also the case for metformin and for amoxicillin, that these concentrations were much lower than the concentrations that we found in the milk. So indicating that the exposure in the at least for the compounds that we tested in the piglets in the system was actually very low, and that can give you some guidance in terms of safety of these compounds. I think one of the most interesting results that we got here is we calculate the milk to plasma ratio in the animal study for those 4 compounds that have been tested in vivo in the Göttingen mini pig, and compare that to the reported milk to plasma ratios in human from clinical lactation studies, that we found a very nice correlation. I think we can probably say that we have been a bit lucky. It's only 4 compounds, but still I think it's very promising about the capabilities and the predictive performance of this animal model. There's some more information about the metabolite of venlafaxine, which is the O-desmethylvenlafaxine. It's an active metabolite. Just to mention too that the in vivo model also has the advantage that also metabolites can be covered in the evaluation. Okay, that brings me then maybe further to the in vitro model to tell you a little bit more about that one. I think maybe I'll need to speed up a little bit in time. I don't know, Marjolein, how I'm doing time-wise.
I think you're good.
You're okay. Yeah.
Okay, yeah. Thank you. So for the in vitro models, essentially the hypothesis where we started from is that that we considered that the blood-milk barrier, and actually especially these mammary epithelial cells, are the main barrier the determining barrier for, the determining factor, if you want, for compounds to cross into the milk or rather stay into the plasma. So that's why we decided to try to make cultures of these mammary epithelial cells, and it was after a while quite successful. And we were actually able to do this, and it's not unimportant in the context of this project to do this for human mammary epithelial cells, but also for the mini-pig mammary epithelial cells. And the original objective there was, well, if we can do this also for the mini-pig, and we have high resolution in vivo data for the mini-pig, and we have the in vitro mini-pig model also, that would allow us to much better cross-validate and to let's say, validate the what we call the IVIVE, the in vitro in vivo extrapolation algorithms. We also are still in the process, and we are actually collecting the data as we speak to enhance the biorelevance of these in vitro models by, for instance, using diluted milk as the medium on one side, of course, of the cultured cells. These are just some background characterization data. This graph just indicates that if you keep these cells in culture, you have to have a little bit of patience for about 1 month, that actually the electrical resistance across this barrier is increasing, this transepithelial electrical resistance, which is an indication for the fact that these cells also in vitro manage to form a biological barrier that has resistance to transport of ions, and also will then represent the biological barriers for compounds that you put on them when you want to measure the transport. Also the same for the mini-pig mammary epithelial cells. There, the nice thing is that for the mini-pig cells, you were able to achieve this functional model in about a week, where it took up to 4 weeks for the human model. So that's, of course, also a species difference that we had to deal with. I guess the most important aspect here in terms of the project that I'm discussing today is that if we looked at the permeability coefficients of about 10 model compounds with diverse physicochemistry in the human mammary epithelial cell model, we did see quite a broad range in permeability coefficients. So we have more hydrophilic compounds showing much less transport, whereas smaller and more lipophilic compounds, you see that venlafaxine also shows up here again at the higher end. The one that was accumulating in the milk also has a higher permeability coefficient in this in vitro model. Of course, you have to also consider the limitations here. I think it was also the message that Michael was bringing before for the placenta. This is just a very simple in vitro model. Of course, these cells are there, but that's the only resemblance that you have with in vivo. So you have to translate this kind of information and combine that with in silico approaches to translate that into a decision or a prediction that you can make for the human situation. Also for the, and that's not surprising, I guess, for the mini-pig mammary epithelial cells, you're glad to see that we did see a comparable kind of range of permeability values, so that we were able to distinguish between compounds that showed higher versus lower transport across these biological barrier. We're still in the process of processing these data, implementing them in the PPK model, but we are quite hopeful that feeding this biodigital twin concept, the PPK model, if you want, with these in vitro data, will have an advantage in terms of predictive performance towards the human situation. So I talked about the in vivo animal model. I talked about a little bit about the in vitro cell culture model. I do need to give you a little bit more information about what we did in terms of PPK modeling. So what is PPK modeling? And this is, again, the same concept as what was referred to previously as the digital twin. Essentially, you combine physiology, pathophysiology of a human being, of a specific population. It can be an infant, it can be a mother during lactation, with pharmacokinetic properties, call it ADME mechanisms. To what extent is the compound absorbed, distributed into certain tissues and also in the milk? And that allows you to predict in a bottom-up way, based on de novo principles, first principles, what is the concentration time profile of these compounds into the blood, and even also into tissues or into other body fluids, including the milk. This is one of the oldest schemes that I know for PPK modeling, but still a very valid one. Essentially, a PPK model consists of two components. You have a system component, which is the physiology or the pathophysiology. This is all the organs that you have, the weight of these organs, to what extent is blood going to these organs, for instance, and then also the drug-specific components. These are the pharmacokinetic properties and also the physical chemistry of the compounds. Together, that forms a PPK model. And the nice thing is that you can then study, having such a PPK model, you can study intrinsic and extrinsic factors, and what is the influence of those factors on the actual pharmacokinetics. An example of an intrinsic factor is there a state of pregnancy or lactation? That means that actually your physiological component will change, and you can study what is the impact on the outcome, the pharmacokinetics that you will predict. You will see that there's numerous applications already nowadays for PPK modeling. Predicting the pharmacokinetics in the milk during lactation, and also the pharmacokinetics then in the infants during lactation, is one of the major applications that we have been dealing with. Before I go into those details, let me just also mention that one of the advantages of PPK modeling is that you can tune the resolution to which you include certain mechanisms. One of the typical examples is that at each organ level, you can decide whether you adopt a permeability limited model versus a perfusion limited model. To what extent is it actually the blood flow, or is it actually the penetration of the compound in the tissue that is going to be rate limiting in your overall disposition process? And of course, it also means that you have to have that information, but it can make this approach very powerful. This is the general workflow that we followed for a PPK model specifically for the lactating population. You always start from let's say a standard, a reference PPK model, also where you have a lot of clinical data available for verification. I mean, it's still a bottom-up approach, but you will build these models for healthy volunteers, and then you will extrapolate it by changing the physiology, for instance, to the maternal, the lactation PPK model, or to the pediatric PPK model. And that can be children of different ages, and depending on the population you are interested in. To make a very long story short, what you can do, you can actually calculate what is the concentration that you find in the blood of the child, and you can also calculate what is the dose that you have delivered during maternal taking of maternal medication, what is the dose that is delivered via the milk to the child. Of course, making certain reasonable assumptions, what is the volume of milk that is taken on a daily basis per kilogram body weight, to give just an example here. In terms of PPK model performance, we went through these different steps through this workflow. This is just an overview when we compared the predicted peak concentrations with the observed peak concentrations for about 10 model compounds, and you see each dot is actually another study and another drug. That in terms of predictive performance for the reference models, we were doing very well. Also for the exposure, you see here AUC instead of peak concentration, that's the right panel. This was also satisfying. And then we actually extended these models to the lactating population. In each case, you can see the point, which is the reference value, what was measured in a clinical lactation study. Or actually, the point is what was predicted. Sorry about that. The point is what was predicted. And then the range is what has been reported in clinical lactation studies. You will see that, because of the limited information available from the clinic, sometimes that's a very broad range in the milk to plasma ratio. But in most cases, we were actually hitting the range that was also reported based on clinical studies. Of course, these models need to be further optimized, and we also need to improve the genericity of these models, and because now we have built compound-specific models, but at some point you want to gain sufficient confidence in this type of PPK modeling framework that you can apply it for every new compound, even when there is no clinical lactation data available. These data also allow you to, as I quickly mentioned before, to calculate the relative infant dose, which is a relative indication of the percent of the dose that is reaching the infant relative to the maternal dose. And you see that for most drugs, and in a way that's actually known, but not for all drugs, but for most drugs, these relative infant doses are very small. Of course, then you still have to assess to what extent is this potentially still an issue, a safety issue in the infant. But the nice thing is that you get quantitative information that can help support your decision-making regarding safety during safety of medication during lactation. That brings me to the conclusions. So the clinical lactation studies, what I mentioned in the beginning, of course, yield unique quantitative data regarding concentrations of medicines in human milk. Even with a very limited number of measurements, this can be a game changer, especially when combined with PPK-based simulations, what I presented in the latter part. I think we also can conclude that the developed PPK models resulted in reasonable prediction of human milk concentration time profiles for at least 80% of the tested medicines, and we are improving this further as we go along. Then we also have the in vivo animal model that I presented, which is based on the gutting and minipig, and it seems that it may represent the gold standard species in case of in vivo lactation studies need to be done before in a preclinical stage. So the milk to plasma ratios for the 4 compounds that we have studied correlated very well with the milk plasma ratios that were reported previously for the human. The in vitro model, I think there's still also some work to be done there, but the nice thing is that we were able to distinguish between low and high permeability coefficients for these different model compounds. Of course, these in vitro and vivo algorithms need to be developed further, and we need to see how we can use these permeability data, permeability coefficients as input in the PPK models. At least we think that within IMI conception, we have pulled this this domain forward and actually started to make a proposal to generate a proof of concept so that it's possible to generate quantitative pharmacokinetic data specifically in these populations, and that in a very standardized manner. Before I end, I just want to very quickly then cover what is next. So we have already a name for a follow-up project, a follow-up consortium, if you want. It's called Milk for Baby. You see the different names of the partners that are already there. This is very preliminary. I want to add that, but where we want to focus really on recruitment and sampling. So to what extent is it possible to recruit patients and to be able to access samples, and because when we have these samples we can generate this information. So this is about the clinical lactation studies. How do we store them? We need protocols for that, and that needs to be further standardized, and there also a network covering these efforts needs to be established. We need to further focus on the bioanalytical aspects according to the highest quality standards, also the pharmacometrics aspect, modeling, whether it's top-down or bottom-up modeling. All these technologies that are advancing very rapidly needs to be leveraged. And of course, it's very important to then disseminate that information, go into interaction also with regulatory agencies, something that we have also done for the non-clinical methods recently, and see what we can also learn from there. And that's also very important. I have some further text about Milk for Baby, but maybe with respect to time, it's better to give it a little bit of time now for questions. And I thank you for your sincere attention.